This video will provide you with an overview of DNA replication. We'll briefly recap template-based DNA synthesis and then discuss the three major topological challenges associated with replication. What lessons have we learned so far about the structure of DNA? The structure of DNA is a double helix. This secondary structure provides the basis for semi-conservative replication. The complementarity between the two strands offers a straightforward concept for DNA replication. To create a new copy of a DNA double helix, each parental strand serves as a template for synthesizing a complementary strand. This results in a new DNA double helix that contains one parental strand and one newly synthesized complementary strand. This process highlights that DNA synthesis is likely unidirectional due to the inherent polarity of the DNA strands themselves. DNA replication needs to be both fast and accurate to ensure the faithful transmission of hereditary information between cells and across generations, while also allowing for rapid cell division. The following data demonstrate that DNA replication is indeed both accurate and fast. In E. coli, the rate of DNA replication is 500 nucleotides per second, while in mammalian cells, it's 50 nucleotides per second. This difference in replication rates is likely due to several factors, including the varying rates of cell division between prokaryotes and eukaryotes, the necessity for greater accuracy during the replication of large eukaryotic genomes, and the presence of nucleosomes in eukaryotic chromatin fibers. Despite the difference in replication speed, the accuracy of DNA replication is similar between prokaryotes and eukaryotes. For instance, the genome of a mammalian cell is about 3 billion base pairs, and the mutation rate per round of replication is 1 in a billion. This means that for a gene that consists of 1,000 base pairs, the mutation rate is 1 in a million. This high level of accuracy ensures that genetic information is reliably transmitted with minimal errors, which maintains the integrity of the genome across generations. Now let's discuss the mechanism of DNA synthesis. DNA synthesis cannot occur without a primer, a short oligonucleotide that recruits DNA polymerase to the DNA template. The primer anneals with the template strand, and its orientation determines the direction of DNA synthesis. DNA synthesis is catalyzed by DNA polymerase, which binds to the primer that's attached to the template DNA. One of the functions of DNA polymerase is to catalyze the formation of a 3' to 5' phosphodiester linkage between the 5' phosphate group of an incoming deoxyribonucleotide and the free 3' hydroxyl group at the end of the newly synthesized strand. The DNA polymerase reads the sequence of the template strand and catalyzes the addition of complementary nucleotides at the 3' end of the newly synthesized strand. The formation of the phosphodiester linkage results in the release of pyrophosphate. Pyrophosphate is quickly degraded into two inorganic phosphates. This drives the equilibrium toward DNA synthesis and away from DNA hydrolysis, which ensures efficient DNA replication. One consequence of DNA structure for replication is that the two strands must be separated, which leads to a replication fork forming. The replication fork and DNA synthesis move in the same direction. Since the two template strands of DNA are antiparallel, but DNA synthesis is unidirectional, only one strand can serve as a template for continuous DNA synthesis. This strand, known as the leading strand, is synthesized continuously and elongates toward the replication fork. The other strand, the lagging strand, serves as a template for discontinuous synthesis. The lagging strand is built from Okazaki fragments, which are short segments that are synthesized away from the replication fork. These fragments elongate in the opposite direction and are later joined to form a continuous strand. This joining process ensures that both DNA strands are replicated accurately despite the unidirectional nature of DNA polymerase. The double helix structure of DNA presents several topological challenges during replication. One of the primary challenges is how DNA can be replicated without a total separation of both strands. DNA replication initiates at specific sites, known as origins of replication. In circular DNA molecules, such as bacterial genomes and plasmids, there's usually a single origin of replication. This origin gives rise to a replication bubble with two replication forks that move in opposite directions. In contrast, linear eukaryotic chromosomes have multiple origins of replication along their length. Each origin similarly forms a replication bubble, with two forks moving in opposite directions to ensure efficient and complete replication of the chromosome. These specific features of DNA replication lead to three major topological challenges. First, as the DNA strands are separated, there is a local change in the twist number, which causes DNA supercoiling. This supercoiling must be managed to prevent tangling and ensure smooth progression of the replication forks. Second, when replication nears completion, the two replication forks moving in opposite directions will eventually meet, potentially leading to the interlocking of the newly synthesized double-stranded DNA molecules. These interlocked DNA molecules must be carefully separated to ensure that each resulting cell receives an accurate copy of the genome. Let's examine the first topological challenge, also known as the swivel problem. When the DNA strands separate at the replication fork, there is a decrease in the twist number, the number of turns one DNA strand makes around the other, without a change in the linking number, the number of times a strand of DNA winds around the helical axis. Since the linking number remains unchanged, the writhe number must become more positive, which leads to the formation of positively supercoiled DNA ahead of the replication fork. This accumulation of positively supercoiled DNA can slow down DNA replication. This swivel problem is resolved by enzymes called topoisomerases, specifically topoisomerase 1 and topoisomerase 2. Topoisomerase 2 functions by temporarily breaking both strands of a DNA double helix to pass another segment of the DNA through this break. This mechanism changes the DNA's linking number in increments of plus or minus 2 and modulates the writhe, not the twist, of the helix. These negative supercoils favor the unwinding of the DNA double helix, which ultimately facilitates the opening of the replication fork and promotes efficient DNA replication. The second topological challenge arises when replication forks moving in opposite directions meet. This scenario is illustrated with the replication of a circular DNA molecule. As the two replication forks approach each other, the double-stranded DNA between them experiences topological constraints, which leads to supercoiling and interlocking of the newly synthesized DNA molecules. There are two ways to solve this problem. The first option is decatenation before full replication. In this approach, topoisomerase 2 acts before the complete replication of the DNA molecules. It cleaves the two unreplicated parental strands, which allows the partially replicated molecules to be separated. Following this decatenation, DNA synthesis is completed independently on each circular DNA molecule, resulting in two fully separated and fully replicated circular DNA molecules. This process is depicted on the right side of the diagram. The second option is decatenation after full replication. Here, DNA synthesis is completed first, resulting in two fully replicated circular DNA molecules that are interlocked. Topoisomerase 2 then acts to decatenate these fully replicated molecules. This means that the two circular DNA molecules are separated only after the replication is fully completed. The key difference between these two options is the timing of decatenation relative to the completion of DNA synthesis. Decatenation can occur either before DNA synthesis is finished or after it is fully completed. In both cases, topoisomerase 2 plays a critical role in resolving the interlocked DNA molecules to ensure proper segregation and function of the newly replicated genomes. The end of DNA replication in eukaryotic chromosomes represents a third topological challenge. The ends of eukaryotic chromosomes contain multiple repetitive, non-coding DNA sequences called telomeres. Telomeres are located at the ends of eukaryotic chromosomes. The enzyme telomerase plays a crucial role in replicating these regions. The end replication problem arises because leading strand synthesis fails to reproduce the last part of the telomere, leaving a blunt leading end telomere without its characteristic and crucial 3' overhang. Telomerase is a ribonucleoprotein particle that contains an RNA component, which serves as a template for DNA synthesis and a DNA polymerase component that belongs to the RNA-dependent DNA polymerase family, also known as reverse transcriptases. This means that telomerase can synthesize DNA using its RNA template. Let's go over the role of telomerase in eukaryotic DNA replication. After the removal of the RNA primer from the last Okazaki fragment, the 3' end of the chromosome, corresponding to the leading strand, has a 3' overhang. This overhang is longer than the 5' end of the chromosome. Telomerase extends the 3' overhang by adding multiple repeats to the leading strand end. This extension process is catalyzed by the reverse transcriptase activity of telomerase, which uses its RNA component as a template for adding DNA repeats. Once the 3' overhang is extended, a new Okazaki fragment can be synthesized, extending the lagging strand. The extended 3' leading strand now serves as a template for this process. After the RNA primer of the last Okazaki fragment is removed, a new 3' overhang is formed. However, because of the prior extension of the 3' end by telomerase, there is no loss of genetic material. This process ensures that the chromosome ends are fully replicated without losing crucial DNA sequences. Ultimately, telomerase ensures the maintenance and integrity of chromosome ends and prevents the gradual loss of genetic material that would otherwise occur due to the end replication problem. This is essential for the longevity and stability of eukaryotic cells, especially in rapidly dividing tissues. Let's delve into how telomerase functions, using the human telomerase as an example. When DNA replication reaches the end of a eukaryotic chromosome, telomerase attaches to the 3' end of the leading strand. In the enzyme shown here, the RNA component is depicted in light green. The telomeric repeats in human DNA are sequences of TTA, GGG, TTAG. The RNA template within telomerase contains a complementary sequence, AAU, CCC, AAUC, which allows for base pairing with the DNA. The last part of the DNA repeat, TTAG, pairs with the first part of the RNA template, AAUC. This pairing positions the telomerase for extension. The enzyme then extends the 3' end of the DNA by adding complementary nucleotides, which creates a new DNA sequence, GGT-TAG, from the RNA template CCA-AUC. The end of the newly added DNA segment, TTAG, is complementary to the RNA template sequence AAUC, which makes the reannealing of the telomerase that has dissociated from its original position possible. Therefore, the telomerase can sequentially elongate the telomere. By extending the 3' end of the leading strand and enabling the formation of new Okazaki fragments, telomerase prevents the loss of genetic material that could otherwise occur at the chromosome ends during replication. In conclusion, the structure of the DNA double helix provides a straightforward mechanism for understanding how genetic material is duplicated and inherited across generations. However, it also presents several challenges during the replication process. The major topological challenges associated with semi-conservative replication stem from the partial opening of the DNA double helix. During replication, strand separation creates positive supercoils ahead of the fork. Topoisomerases 1 and 2 remove these supercoils to enable DNA replication. When two replication forks converge, interlocked DNA molecules can form, which hinders genome separation. Topoisomerase 2 resolves this by cutting and untangling the DNA in a process called decatenation. Topoisomerase 2 resolves issues by removing positive supercoils, introducing negative supercoils, and facilitating the opening of the replication fork. It also addresses the end replication problem by untangling DNA and ensuring proper chromosome segregation. These topological challenges underscore the intricate mechanisms that are involved in DNA replication to ensure accurate and efficient transmission of genetic information from one generation to the next. Understanding and addressing these challenges are crucial for maintaining genome stability and cellular function.